Okay, hello. I'm Robert Bain. If you don't know me, I'm kind of into computational neuroscience, AI, cognitive neuro, all that jazz. And I'm really interested in recently the history of where neuroscience and artificial intelligence intersect. And I'm going to try to show you guys some of the stuff I've learned about why they are actually called artificial neural networks. Okay, one of the oldest assumptions the field has right now is just how an individual neuron, a real neuron, as shown on the right, is abstracted into just computations. And if you're not familiar with this on the left, let me walk you through it. The X's with different subscripts will be your different inputs. So you have input 1, input 2, input in, and each one will be multiplied by a scalar value, and this is what they refer to as a weight. And so these are just linear weights, and we'll search for these, as you'll see later, with something like backprop. Back in the old days, they would just hand tune these weights. So once you've got your inputs and you've multiplied them by their respective weights, they are summed. And then an activation function is applied. A lot of the times the activation function you'll use nowadays is ReLU. This can be interpreted kind of as once a certain threshold is reached at post-summation, the value will be non-zero and you'll actually output something. Okay, and so I've kind of color-coded things to be the same on the right with the real neuron. Inputs in the real deal will come and arrive at the dendrites. And if a threshold signal is reached and the activation function triggers, that type of mathematical abstraction on the left actually occurs at the trigger zone on the right. And so if the threshold of voltage across this cell membrane is achieved, and we'll go over that in a second, then an output is sent along this axon to its neighbors to then be received by its neighboring neurons' dendrites. Okay, I'm going to talk about voltages a lot, and how the voltage is measured is just the difference in electromagnetic force, right, between the outside of the cell and the inside of the cell. And that is demarcated by this bilipid layer shown here. And there's different concentrations of charged ions, charged chemicals, inside and outside the cell, and they're kept that way because the cell membrane is impermeable to them diffusing across it. Except for the fact that there are certain proteins embedded, transmembrane proteins, that can be selectively opened or closed to allow or to forbid certain chemicals from going across their chemical gradients and going through the cell membrane to the other side. So one way that we need to be aware of how these receptors selectively open or close is by ligand binding, which just means some chemical is going to come and interact with a certain site on the protein or the channel receptor, and it's going to change its shape such that it opens or closes. So here, that's what this blue pentagon has done. It's going to be here for just a little bit, long enough to change the shape such that sodium can pass through its concentration gradient from outside the cell into inside the cell. The other way you can open these or close them, respectively, is they can be sensitive. Their shape can also just be sensitive to the voltage. So usually they'll have some charged part exposed to either inside or outside the cell. That's actually part of the amino acid sequence that makes the protein, and it can be sensitive to the voltage and thus change its shape, allowing it to open or close as well. Okay, and so this point neuron model from 1907 was, I've heard people cite different things, how far this point neuron model goes back, but Louis Lepic is the oldest one I found, and he is a French neuroscientist. And it assumes that the 3D structure of these dendrites and axons in the real neurons can just be ignored and replaced with computations that are linear. And it also assumes that the only action potentials are voltage spikes. So potential and voltage are synonymous, so you can think of action voltages, what have you, or just spikes as you'll hear them. A spike over the history in neuroscience has been beaten up and changed just a little bit, so it gets confusing, but really it is just an inflection point where something changes in the summation dynamics, and you see a sharp demarcation and a change. But what always happens though is you'll see the voltage as we record it over time goes up and then comes back down, and often that up and back down is rather dramatic, as we'll see. And so yeah, the Peaks model, the point neuron model, assumes that the only place that produces such a spike in voltage is at this trigger zone here, not anywhere else. And both these points, as we'll go into later, we know are really wrong assumptions. And so I added an extra neuron here and showed you if this is the trigger zone of this smaller neuron and it has decided to spike, we would measure its voltage as x1, and then we know the voltage is going to be transformed to something else by the time it reaches this trigger zone to be summed, and we're going to call that y. And so it's called x1 because this is an analog. This is what we're usually abstracting over here in the point neuron model, and w1 is going to be this linear weight, this w1 over here. And so x1 times w1, it's always the same w1, regardless of any other previous firing or inputs. And we'll learn later that this, assuming that x1 times w1 will always get you y in the real neurons, this is not a good assumption either. So history point 2 we'll need to know is Rosenblatt's perceptron. So we've kind of fast forwarded 5 decades here to the 50s. And Frank Rosenblatt was a neurobiologist at Cornell eventually. So he was really interested in the brain, and he got famous for his perceptron, which tested a specific neuroscience theory that learning in our brain happens by changing the connection strength between neurons. And we can see here kind of what his first perceptron would have looked like. There were 400 inputs on the left, and so he had a 20 pixel by 20 pixel camera, so that's where the 400 inputs come from. And each one of these circles here, each node or neuron, its original input value would be some scalar value, which would be the intensity of the photodiode in the camera at a particular location in the photo image. And then each neuron had one weight connection to its neighbor, the single neuron in the output layer. And he came up with a clever way to learn and to update these weight strengths such that over time it could answer yes or no questions really well. So I don't remember this particular yes or no questions he was asking, but nowadays, if you're familiar with computer vision, you could imagine, hey, I want to know, is there a cat or is there a dog in this image? Things like that. You could imagine asking 2 yes or no questions instead and have a second output neuron added, and it's going to get its own weights as well, connecting to the input nodes. And this starts looking a little bit more like the deep neural networks, multi-layer perceptrons we know nowadays. Well, they're not MLPs yet, multi-layer perceptrons, but we're about to get there. By the time 1986 came around, a whole bunch of cognitive scientists were using artificial neural networks to try to explore how cognition works in the mind. And they were working with Frank Rosenblatt's perceptron, but more often they were they'd already inherited a multi-layer perceptrons, which is shown here on in the middle figure. And the difference between a single layer perceptron and a multi-layer perceptron is you'll have hidden layers. And the reason they're called hidden layers is because we can't pre-subscribe before training, and often it's impossible even after training, to figure out a human label, a human symbol for just what these neurons mean when they're firing or not. And that's in the contrast to the input and the output neurons. We can very much say what they are, and we can say the input neurons are intensity values of a photodiode in the image captured by a camera, or in the output's case, it's we're going to train for some yes or no question. But if it's in the middle, if it's in between those, we call it hidden. And these were essential to do even simple logical gate-like operations like XOR. Marvin Minsky and others were famous for pointing out that Rosenblatt's ambitions and wants for his perceptron probably couldn't be met if his perceptron couldn't even do simple XOR. And so that's why these hidden layers were added. Okay, part of this parallel distributed processing group in 1986 included Geoffrey Hinton and Rummel Hart and Ronald Williams. And maybe Hinton especially is most famous for introducing backprop, or specifically learning representations by backpropagating error. And I won't go over the equations, but I can give some intuition about the calculus behind these update rules. And what backprop does is it allows you to provide labeled data. So you would provide a whole bunch of pictures with cats in them and without, and you would need to tell the network after it guessed whether it guessed correct or it guessed wrong. And then using that data, you can update the weights, and if you're lucky, you'll have learned something interesting. And so here's a really simple artificial neural network on the right. This is too simple to be real, but it's harder to add more weights because visualizing it is hard. But the bottom plane is going to be the configuration space, or the weight space of the neuron, or the neural network rather. So you can imagine this white dot, this white star rather, is where we currently are, the current configuration of the artificial neural network we're trying to train. And its error is a little bit high, and that's going to be the z-axis. And you would never, during training, produce this whole image, but what you would do is you can use calculus. You can use the chain rule of calculus iteratively over all the different layers of your network using that labeled example in your artificial neural network's guess. And you can calculate a very small portion of it drawn in the circle. And you can get some physical intuition for this. And how learning happens is you can imagine that you are this white star, and there's a whole bunch of fog around you, so you can't see this valley-like structure very far away. But what you can see is you can look at your feet, and you can try to go, well, I'm trying to get downhill where the error is really low, and so I'm going to look at my feet and just see which way's going downhill the most. I'm going to follow it for a little bit, and then you'll get to some place and you'll reassess, and you'll do this again. So ultimately, assuming you're not stuck in some local optima, you'll get pretty close to a good solution, if not an optimal one. And so that's how this backpropagating errors works at a high level. And so given these 3 points of history, they actually get you quite a far way into the bag of tricks we've inherited in deep learning. And I'd like us to really look at this oldest assumption from Lepeque's point neuron model. I'm going to try to present as much evidence as I could find in the last couple weeks about the 3D structure being abstracted away and replaced with just linear equations when that is not appropriate. And then we've also discovered there's a lot more action potentials, a lot more places where spikes occur than just at the axon hillock. Okay, and I'm going to be talking almost exclusively about one of the most complicated neurons in the mammalian brain, pyramidal neurons. And these are abundant in places that we associate with advanced cognition. So to be able to figure out how to model these would be really cool. And then hopefully we could be able to make mathematical abstractions and trade off some of the fidelity of our models for the things we're really interested about, just just the bare bones of computations we'll need to produce intelligent AI systems. And these pyramidal neurons come in several different flavors. We'll see some that aren't in the neocortex, but as you can see, we're zoomed in on a part of the neocortex right now. The neocortex is a large portion. It's 3/4 of the volume of our brain. Most of what you're seeing on this bottom left image, except for maybe the cerebellum and some brainstem, this is all neocortex. And it's classically got this 6-layered architecture in it. Layers 2, 3 tend to be combined, so you'll see this sometimes. But these pyramidal neurons are found in every single layer. The biggest ones are layer 5 pyramidal neurons, and that's because their cell body, or their soma, is in layer 5. And their cell body is what gives them, the shape of their cell body is what gives them their name, pyramidal cells or pyramidal neurons, because they're kind of shaped like a pyramid. The axon of these pyramidal neurons isn't shown here. They're truncated. They often would shoot straight down, these ones. So really what we're seeing is the cell body and then all of the dendrites. So the dendritic tree or dendritic arbor is really extensive in these pyramidal neurons. And they come from both ends. There's this little bush at the bottom, and then they extend all the way up into layer 1 sometimes. There's also layer 2, 3 pyramidal neuron cells and layer 6, and there are some in other parts of the brain as well, as we'll see later. Okay, so that bushy bottom bit of dendrites near the cell body is called basal dendrites. A little further up, well, I guess this blue is the apical trunk, the apical dendrites. And then before you get to the tufts at the very end, and they kind of bifurcate here, you've got these offshoots called oblique dendrites. And they all have different inputs and different properties, as we're going to go over a little bit. But if you're not familiar with these, the naming convention really threw me through a loop. So I came up with this cute little acronym, or I came up with this cute little way of remembering mnemonic, BOAT. So, B stands for basal, and it's the first letter in BOAT, so it's going to be the dendrites that are closest to the cell body. In comparison, T is the last letter in BOAT, so tuft is going to be the farthest away bits of dendrites with respect to the cell body. And in this diagram, the axon here shooting off the bottom, which could be very large, is cut off and not shown. And this figure here on the right by Spruston in 2008 is meant to show all of the different computational domains we have some evidence that exist. So to be able to abstract away this neuron, you're going to need probably like 6 little different equations as opposed to the 2 we have or 3 we have with the point neuron model. And they have, each of these compartments are like, they'll have either different inputs coming from different, neighboring structures. Like we saw layer 1 was really far away in the L5 pyramidal cells, so they might get different inputs than down here at the cell body. The way the inputs are integrated can be different in these different colored domains. The branching patterns are different, and branching is a major source of voltage attenuation, so that matters as well. The protein channels or the protein receptors embedded in the cell membranes can be different at different locations. Their densities can be different, even if they are the same type in one location as relative to the other. And these all affect the different computations that are possible in these distinct domains. So I'm kind of jumping to modern day understanding. On the right, we have active dendrites, which represents more of our modern day understanding. But on the left, we have an example of what we used to think was going on, especially pre-1990s. We thought that these dendrites could be modeled as passive cables that just filtered the voltage as it traveled further distance. You can imagine a wave breaking on the ocean, and it might initially be rather tall, but as it travels further and further, it smooths out. And the height of that wave is a good analogy for the amplitude of the voltage wave as it is received in the dendrites and then is attenuated as it gets closer to the cell body. And that's what we're seeing here on the purple on the left. And we know that instead of being passively filtered, as that wave of voltage reaches the cell body to be summated at the axon hillock, instead we know there are actually a bunch of different ways that that voltage can be regulated, upregulated, downregulated, and that's achieved by these different spikes that we know exist. And so now, instead of calling them passive dendrites, we know that there are active electrochemical processes that can change that voltage as it travels. And so we call dendrites now active dendrites. This is almost a certainty. And you see we have sodium spikes, which the normal spike we actually do capture in our point neuron model at the axon. That's a sodium spike. So, but we've also got some of that sometimes in the dendrites. We've got two flavors of calcium spikes. One is longer duration, so it's more of a referred to as a plateau. And then there is, this one can get complicated, but it is called an NMDA spike. And it is shown here as being lasting 100 milliseconds. We'll talk later about how this can actually last much longer. Okay. And just here's a figure from Larkham et al. 2009, and this shows where these different spikes can actually be initiated or thought to be initiated. So here, like I said, that axon spike is the sodium spike we know and love. Up here near the bifurcation zone at the top of the apical dendrites where they become the tough dendrites, we've got a hot zone for calcium initiation. And then in the furthest the furthest thinnest dendrites, they all, or not all, but they often are where NMDA spikes will be generated. Okay, and we have both simulated on this slide and on the next slide real experimental data that shows that maybe we could have known passive dendrites weren't going to be a thing. Right? Or at least now in rather retrospect, we know that they couldn't have ever been a thing. And let me walk you through this simulated hippocampal cell and show you that intuition. And it's worth noting, here's the hippocampus on the right. You have to cut away and get underneath the neocortex to see this bit of allocortex. So it's a 4-layered structure instead of a 6-layered structure. And it deals with spatial navigation and with turning short-term memories into long-term ones. Okay, so let's see. So here where this arrow is drawn in A, you'll see that a lot of these dendrites around this arrow that are far away from the cell body are this dark purple. And if we look at the figure here, we see this means 0.013 voltage. And this is the voltage measured at the soma, at the cell body, if we electrically stimulated it here. So we'll use the same type of electric stimulation all across the stimulated neuron, and then we're recording at the cell body what voltage it is by the time it travels there. And these don't have, the simulated neuron doesn't have any of those active dendritic spikes like we saw on the last slide. This is what we would expect if they were just passively filtering the voltage. And this is a logarithmic attenuation of the voltage. Something that, we really could just use backprop to search for a linear weight for, right? But, okay, so the voltage is very small, especially when you're far away in the dendrites, when measured at the soma. But figure B here shows what the local voltage is. So it's the same type of, we're going to stimulate at all these different locations using the same kind of electrical shock, but instead of measuring at the cell body, we're going to be measuring the voltage kind of really just close to where we're actually stimulating it. And so even though both the arrows in A and B, where we stimulate, the local dendritic voltage is 14.2. By the time it travels all the way to the cell body, we know it's only 0.013 volts. And so The point here is that the voltage in the distance tuft is attenuated 900 times by the time it passively reaches the trigger zone. And I've heard people like folks at Numedta, Subotai, and Jeff Hawkins talk about 90% of the connections in a neuron just being wasteful if you really think that the point neuron model is a good approximation. And this is kind of what they're talking about. A typical cell in CA1, the hippocampus region CA1, like shown here, would have 30,000 connections. And if it were only passively propagating this voltage, the majority of them are going to be too far away to really make an impression at the cell where the spikes fire. So they've really got a point there. And so that last slide showed simulated data. Here we've got the real deal from Major et al. In 2013. And so what we're going to do here at B, C, and D is we're going to apply sugar. So sugar here, and then sugar is here, D sugar is here. And with B, we're recording at this blue, this spot, this blue electrode, and we're just measuring voltage. And we give it a puff of sucrose just a little upstream, up here, and then we're going to record. And you'll see, the y-axis is only a couple millivolts, so not too large of a signal is being generated by the sucrose. But just a couple hundred micrometers downstream of the blue electrode, we see at the bifurcation zone where that calcium hotspot is, almost none of this signal's really getting picked up. You can kind of correlate like this big spike to a spike here, another big spike here. But given that the y-axis is 2 millivolts, this is very, very small. So where the sugar isn't enough to initiate those any of those other spiking dynamics that we were talking about, NMDA spikes or calcium spikes. And in this really small regime and really small inputs, it's not getting very far before the voltage is just filtered too much to be meaningful. Even at just the next trigger zone, not even the soma, the cell body, where it's really going to matter. And then C shows a very similar picture. But we're going to put sugar here, measure at the black electrode here, and then measure at the soma. The black electrode picks some stuff up. Notice this is actually a slightly smaller scale than what we just looked at. And then the soma, just like in B, you go a couple hundred micrometers down, and you really can't tell anything's going on upstream. And then D is going to be very similar, but the red electrode is in the basal dendrites. But it's the same picture. Okay, and so we're going away from making the case that passive dendritic cable equations just don't work. You need some active processes. This is an example of an active process that can be invoked, and it's actually invoked and helped by a backwards propagating action potential, or a BAP. And so that's what the slide header means up here in the top left, that they go backwards too. Because, I remember learning even in my undergrad, the sodium spikes that occur in the axon, since a major component of the action potential is due to voltage gated sodium channels. And so what will happen is they kind of once enough are triggered in a density at that axon hillock, they'll kind of propagate down the axon, because since they're voltage gated, when enough open, they're letting in sodium into the cell. They're causing the voltage to go up, and then their neighbors get triggered as well, and it creates this kind of cycle, and it'll travel down the axon to signal to its neighbors at their dendrites. But actually, what we've learned is that these sodium channels aren't just at the axon hillock and in the axon towards the neighbors, but it's also in the apical dendrites and in other bits of the dendrite as well. So this action potential can actually travel backwards into the cell body as well. And they can be combined with your inputs to be to assist in causing active dendritic spikes. And so that's what we're going to see here. The difference between B and C is they both have a backwards traveling component after the cell spikes. But the only difference is here in B, we're stimulating the cell at the cell body to make it spike. We're doing the same in C, but we add just a little bit of electrical shock, a little bit of current at the red electrode after we stimulate here, because there's a little bit of time delay as the action potential here at the soma travels backwards. And so we can see if you don't have this perfectly timed current at the red electrode, there's no spiking behavior. We're just seeing first this soma spike travel backwards. It hits the blue. And then just a little bit time later, it hits the red electrode. But if we stimulate it just right and we make it so this back propagating red wave is met with just a little bit of extra oomph, we get a spiking behavior. So I said a spike is sometimes hard to define, but it's always, the spike is always a voltage going up and then a voltage going down. And it's also where you get this sharp demarcation in summation where the dynamics are obviously changing, right? And just this is a good example where the red before doesn't really shoot up very sharply, but here it does shoot up and it stays up for quite a bit, and then it'll come back down. And you'll notice that this is a calcium spike. This is actually referred to as a backwards propagating action potential induced calcium spike. So it's like BAC, a back spike. Some of these acronyms get kind of crazy, but this is something you really can't account for in a regular neural network. Maybe in a recurrent neural network, but things get a little all combined together in that instance, and it's hard to train with backprop. But you definitely don't get intricate behavior like this in a single point neuron. But you could do in more complicated spiking neural network models. Okay, and you'll see that because this spike, it gets a little, you can get even a little more complicated and notice that this calcium spike here was large enough to actually induce an extra burst of 2 more spikes at the soma. So, there was electrogenesis, there was a current induced here when the BAC fired, and it was strong enough that it actually traveled back down to the soma and induced 2 more spikes. So that's kind of neat. Okay, so I said there were NMDA spikes, and those are made possible by the NMDA transmembrane channel receptors. And what I'm showing you here is that NMDA receptors are a little special because we've talked about receptors that open because they bind to something like glutamate. That's the blue pentagon shown here. And then they can also be sensitive to the voltage across the membrane, whether they open or close. NMDA receptors require both. And so I talked about the sodium action potential traveling down the axon or traveling back into the cell in the case of a back propagating action potential, and how those are made possible by the voltage gated sodium channels opening, changing the voltage, and kind of causing this chain reaction with their neighbors that do the same, and it'll propagate. But because both of these things are required for NMDA receptors, both binding to glutamate and having an appropriate voltage, they tend to be local when you produce them. So this is rather interesting. We didn't account for things like this much before the 90s. And real-time imaging techniques in the 90s allowed us to see and get into these really small dendrites that we hadn't before, and so we could actually do electrophysiology experiments starting in the 90s on these things reliably. And so in the 90s and in the aughts, we really were getting good data on these things finally. So you can kind of see the spatial scale that these NMDA spikes happen in these really small distant dendrites. Usually you're talking about clusterings of inputs and these local spikes happening in 10 to 40 micrometer ranges. You can have as few as 8 inputs cause a spike. And remember I said, I've gone over this twice now, right? But we see from going from 7 inputs to 8 inputs, you get a really big response on this 8 input. So we'll talk about somewhere between 7 and 8 of these spines being activated by uncaging glutamate, that we're getting a much bigger response going from 7 to 8. So there's some spiking, there's some threshold that's been met. And you can actually, it looks to be going over maybe 20, 25 micrometers where where we're stimulating the dendrite. You can have bigger, you can release glutamate over a bigger surface area, over a longer distance, but you'll tend to need more glutamate. And so you could get a 40, 50 micrometer spike going on a dendrite, but you're going to need to stimulate maybe up to 20 spines instead of the 8 needed here. And so here we're going to trigger some NMDA spikes in the basal dendrites just by applying enough glutamate. And once you do get enough glutamate, it's rather binary, the response. You get there's very little range where you get kind of an intermediate response. And this is measuring the voltage amplitude of the following or the induced NMDA spike. And instead of being binary, the relationship between glutamate intensity and the duration of the spike is actually linear. And this is thought to help make the integration time zone of disparate signals, right? So you might have some part of your brain doing auditory processing and some part of your brain doing visual processing, and they'll need to come together, but those tracks and processing might occur at different times. And so you can imagine this is one way to open up a longer window and to wait longer for those signals to actually meet, is to increase or decrease the amount of glutamate that is firing. Okay, so there's coincidence detections, temporal pattern detections, that are facilitated by this relationship. The fact that you can have a stronger input on a dendrite, and then it thus will kind of wait around for synaptic inputs and integrations. And so here's an actual example of coincidence detection. You can imagine going a step further. Once you understand what's going on here, it's very easy to imagine having multiple of these steps combined, and you'll get temporal pattern detectors. But what's going on here is in the very far, far away reaches of the dendrites, we're having strong stimulation that can produce an NMDA spike. But even just downstream, since this is actually a small little chamber with not a lot of volume, can't conduct very much charge, it's got an impedance mismatch, it doesn't reliably travel very far, actually. So that's kind of sad. If instead we don't stimulate up here at the red, but we stimulate at the blue, we're not even stimulating enough to get any type of spiking behavior. This just looks like normal summation. But if you do them at the same time and you space them temporally apart such that they overlap, this big spike in the red that we saw before was enough to induce a spike in the blue. So it's completely changed the behavior of this voltage signal. And combined, they can reliably propagate that voltage all the way down to the cell body and cause a spike down the axon. So this is just to show a simple coincidence detection example, and to say that small dendritic spikes and individual tufts often need to sum with other inputs to evoke spiking at the soma. Okay, yeah, if you held on to this point, gosh bless you. Thanks.